just your choice. Okay. All right, so let me go through these just one by one. Again, I invite you to write this down. Monohybrid crosses, again, you tell me. Does that sound familiar?
Yes.
Okay. So when we say monohybrid crosses, what we mean is a cross between, well, it's a cross showing the inheritance of one single trait, right? One single trait. Now, so when you're completing a monohybrid cross, we've got these potential variations here: complete dominance, incomplete dominance, codominance, and then the sex-linked crosses or sex-linked conditioning. Okay, so let's start with complete dominance. So you do your Punnett square, just a classic four-box Punnett square. Now, when we say complete dominance, what we mean is the dominant condition completely eclipses the recessive condition, right? So whenever the dominant allele, and I'll go through some of these terms in just a moment if they're not making sense, but whenever the dominant allele is present, it will eclipse the recessive. The recessive will not be shown. Okay. So if you've got, and by the way, I'm making this very simplistic today. Some of the characteristics that I'm going to use would not be accurate. Just for simplicity's sake, we'll make it this way. But I'm not suggesting that what I'm presenting at this moment and some of these traits are 100% genetically possible. Okay, but here we go. Let's just say the condition here is capital T controls. We're talking about height here. Here's the characteristic. So capital T means tall, right? And lowercase t means short. All right, it's not that easy. That's what I'm saying, right? It's not like you just get that capital T, you're going to be tall, or you get the lowercase t, you're going to be short. It's not that easy, but just for simplistic sakes. Okay, so here we go. We're going to cross and if these terms don't make sense, I'll explain them. Cross a heterozygous parent with another heterozygous parent. So does that make sense, heterozygous, when I say that? Okay. So each of these letters, each of those letters are considered an allele. That's how I pronounce it. Allele. Now, if we're thinking back to the homologous chromosomes, that means one of these letters, essentially, right? One of these letters came from mom and one came from dad, right? Now, we may know which one came from mom and which one came from dad. In this case, it doesn't really matter. So one of these letters, each letter is an allele. Now, when those letters or alleles are different, and these are different, right? They're controlling the same trait, but they're different. In other words, that capital T controls height, tall, right? It produces an individual that's tall. The lowercase t produces an individual that's short. They're different. They're expressed differently. Okay. So when they're different, we say that is heterozygous. So some of you are saying, yeah, that term sounds familiar. Heterozygous. What's the other possibility? Homozygous. Now, homozygous needs some clarification, right? You can't just say homozygous. What must come after that? Something has to come after you say homozygous. Yeah, you've got to clarify, is that homozygous dominant or is it homozygous recessive? Because homozygous just means those alleles are identical. That's all it means. So if you've got in this situation, if you've got a dominant condition, you've got this, two capital T's, right? And if you've got a recessive condition, you've got two lowercase t's. So this officially would be referred to as homozygous dominant, and this homozygous recessive. Heterozygous doesn't need to be clarified. Automatically, when you say heterozygous, you automatically mean two different alleles. Okay? So what we're going to do is we're going to cross a heterozygous parent, and it doesn't matter if we put that parent at the top, that doesn't matter, or the side. It doesn't matter where you place these. That's up to you. In this case, it really doesn't matter because both of these are identical. And then we just complete the Punnett square. words, when we say complete, right, we're just seeing where do these alleles cross over. Okay, any questions there? Now, the only other step you really need to go to is now looking at the offspring. The offspring are here, these four, potential, right? Yeah, keep that in mind. These are potentials, right? Possibilities. So we look at those and we assess those. And then we have to determine often what's called the genotype ratio or the phenotype ratio. Does those two terms sound familiar? Okay. So say genotype ratio and then phenotype ratio. Genotype means the actual genetic construction represented by the letters. Phenotype means physical appearance. What's the physical expression of that trait? Okay. Let's do genotype first. How would you represent that in a ratio? Now, you can do ratios, you can do percentages, you can do fractions, I don't care, but traditionally this is ratio. How many homozygous dominant do we have?
1 to 2.
1. Good. So we just list it that way. 1 homozygous dominant. Colon. How many heterozygous? 2 heterozygous. Okay, I've got to move down here now. How many homozygous recessive? 1. Now, if your mind works in percentages, and you would rather express this 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive, go for it. It doesn't matter to me. Right? As long as you're correct, I don't care. Does that make sense? Any questions there? Okay. What's the phenotype ratio? What's the phenotype of this individual?
Tall.
This one?
Tall.
This one?
Tall.
This one?
Short.
Okay. So the phenotype ratio is 3 tall to 1 short. Or again, if you want to represent this in percentages, 75% tall, 25% short. All right, any questions up to this point? This is kind of your standard monohybrid Punnett square condition. Okay. All right, if there's no questions, then see, I'm just going to move on to incomplete dominance. Be pretty quick, rapid fire today. Okay. Here, I'm going to leave a few things. You guys need these terms any longer? Let's get that out of the way. Okay, I'm going to keep this the same. I'm going to cross a tall with a short. Again, not this simplistic. Please do not go home and tell your parents that if you get the tall allele, you're tall, and if you get the short allele, you're short. Okay, not that easy. Now, this is incomplete dominance. You understand what it means to be, or you understand a complete dominance expression. How would you, somebody kind of give me a definition. What's going to happen here with incomplete? If complete dominance means the dominant allele eclipses the recessive, what's going to happen here? Okay, all right. So now you're saying the recessive has a little bit more influence, and that is true. Or we might say the dominant has less influence. Any way you want to look at that, okay? So let's complete this. Now, I'm using the same thing just so that you can see the comparisons. Obviously, nothing at the genotypic level, nothing has changed. At the genotypic level, this looks identical to what we just did. Okay, that means actually at the genotypic level, the genotype ratio is the same. The genotype ratio has not changed at all. What about phenotypic? Well, let's do this. What would that expression be? Of that combination, what would the expression be? Tall or short? Tall. What about that?
Yeah, what's incomplete dominance? Short.
What'd you say, Daniel? Medium. That's the answer. What's that? Sure. So here, this is the difference. The heterozygous condition is a blending, a mixture, a combination of the dominant and the recessive, right? So in this case, we've got one tall, two medium height, one short. So again, really with incomplete dominance, the only difference is understanding that this, the heterozygous condition, is a blending, is a mixture, right? Now, in humans, there are some traits, not all, right? Some traits that use this as its primary means of expression, right? Some sort of blending. In plants, it's very often in plants, a blending. In fact, flower coloration, etc., that's often expressed in this pattern, or this pattern or the next one, codominance, one of the two. Okay. Any questions here? Okay. If not, let's go on to codominance. So let me just leave the genotype, phenotype. We're going to use that again. I'll just sketch all that over. Okay, the tall short is not quite going to work with the codominance. So I'm going to give you some different characteristics here. Yeah, let's do it this way. Yeah, let's think of traditionally with flower colors. So let's say that capital R represents red. W, and I'll explain why I'm doing different letters in just a moment. W means what? Okay, anybody want to take a stab at defining codominance? What do you think that might mean?
Both parents have two dominant traits.
Okay, so there's more than one dominant condition. Is that what you're getting at?
Yes.
Okay. Do you think it means that the recessive condition cannot exist? I heard a no, I think. Okay, yes. It doesn't necessarily mean that the recessive condition doesn't exist. It really just means there's competing dominant conditions, right? We say co, we don't say competing. They're both expressed. In other words, they're both dominant, and because of that, they both want their opinions known. Does that make sense? They want to be expressed. They don't want to be blended. That's incomplete dominance. They don't want to be blended. They actually want to be expressed. Okay. So when we have that, we have a situation. So we're still doing the same type of Punnett square, right? This is still monohybrid condition, really. Let's cross a red. Okay, homozygous dominant red, right? Let's cross a red flower plant with a white flower plant. In other words, we would still say these are homozygous dominant. Both of these now could be considered homozygous dominant. Okay, so what's going to happen? Two capital R's, it doesn't matter if I put the R's here or here, that doesn't matter. Now, the reason we use two different letters, again, is because they both want to be dominant. So the kind of classic capital lowercase letter does not work here for codominancy, unless you really do have a recessive condition. Because remember, a lowercase letter signifies a recessive condition. We don't have that, at least in this situation, we don't have that, okay? Now, here's the case scenario. What's happening here? Well, let's do our genotype first. I think that's the easiest. What's the genotype here? Yeah, they're all, what would you call that? They're all what? Yes, all heterozygous. Now, I'm going to keep it in the traditional like ratio form. If you'd rather just write 100% heterozygous, that is totally fine. Not worried about it. I'll just do that, okay? In other words, they're all that way. There's no other option. Now let's think about phenotype. What's the phenotype then of this situation? Is it red? Okay. This is going to produce not a blending. Good guess. It's not going to produce a blending because if they're red and white blended, then we call that what? Incomplete dominance. So that can't happen. I mean, if it did happen, then it would just be incomplete dominance. So in this condition, that can't happen. There can't be a blending. They both want to be expressed. So then you get these interesting patterns. And again, we see this a lot in flower colors, where you get spotted flowers, striped flowers, checkered, polka dotted, whatever you want to call it. You get the idea here. In other words, they're both going to be expressed. So it would be for red and white. Now here, the next description could vary, but say red and white. I'm just going to stick with the spotted because that makes sense. Red and white spotted. In other words, the red and the white are both going to be expressed. And that's going to look like stripes, spots, whatever. Does it make sense? Okay. Young, if you have any questions, let me know. Let me open this door again. Let's get a little bit higher. All right, there we go. Codominance out of the way. Any last questions of codominance? All right, let me erase a few things. What are we doing now? Sex linked. Okay. All right, before we do sex linked, let's think about what we've already learned. How many chromosomes are in the human body? Total? 23 pairs, so 46 total, 23 pairs are important here as well. Very important. So 23 pairs, 46 total. Which pair determines gender? 23rd pair. Okay. Now, when you say sex-linked conditioning, what you mean is this. Whatever trait that is, it's found on the 23rd pair of chromosomes. In other words, it might make a difference whether you're male or female. It might make a difference because if you're female, you have 2 X chromosomes there. If you're male, you only have 1 X chromosome. That means males, if they have a certain trait on that 1 X chromosome, they are much more likely to express the trait. Does that make sense? Females, they have 2 X's there. So even though they may have a recessive allele, it may be eclipsed by a dominant condition on the other X. Does that make sense? And so therefore, something like, see, you've heard of these things, hemophilia, right? Hemophilia is a classic sex-linked condition or disorder. When you hear of somebody having hemophilia, you're thinking which gender? Male. Now, it's not that it's impossible for a female to be a hemophiliac. It's not impossible. It's just extremely rare. Extremely rare. Right? So let's kind of do that situation so that you can see what's going on. Now, there's one added thing we have to do here with sex-linked. Because we have to know if it's the gender, right? We have to know if it's male or female. We've got to show the chromosomes ultimately. So what we do is we just show the chromosomes. Right? Here's the dad, here's the mom. And then we make the allele superscripts. So here's dad, and let's say he is not a hemophiliac. So he's got a capital H there. He's got a dominant allele. He's not a hemophiliac. He does not, there's no expression of trait. He's clear, right? He's fine. The mother is, how would we call that mother? What would we call that mother?
Carrier?
A carrier. Does she have hemophilia? No. So basically this is complete dominance. It's really that mode. But because this is complete dominance on the 23rd pair, we have to express gender, right? So we say heterozygous, that is true. But more, in this case, we're saying she is a carrier. She carries the trait, but she does not have hemophilia, right? Okay. So let's do the cross. Again, we must show the X and Y chromosomes. So I'm just going to do this up here. You can double check. You can do it on your own. So here we got a little bit more. We got a few more things to keep in our minds. Not only do we got to think about is that particular trait expressed, but we got to think is that particular trait expressed in a male or a female, right? Okay. So here we go. Genotype. Let's just do broad genotypes. This is a male or female. What? Female. Does she have hemophilia? She's not even a carrier, right? Okay. This is a male. Does he have hemophilia? No. Female, what is she?
Carrier.
Male, he's a hemophiliac. Right? So the way we express that, because we have to express gender, right? We'll say, because there's several ways you could write this, but I'm just going to pick out the females. I'm going to say, well, actually, genotype you just list it this way, right? So you can just list it this way: one female homozygous dominant, one female heterozygous. Right, I should have pushed this down a little bit. One male, sorry. One male normal. We just say that male is normal, not a hemophiliac. And then one male hemophiliac. Now phenotype, okay, now we got to consider more gender. So we say one female. How would you describe that? One female. What is she? Yeah, one female and say normal. Right? We're assuming normal. And this condition, normal, right? Okay. One female normal. One female. Now, if you want to use the universal symbols for male, female, you can do that this one. One female carrier. Then the males, one male normal, one male hemophilia. So, especially in the phenotype, that's where it gets important that you understand not only who's a carrier, who's normal, and what's the gender. Okay, any questions there? Okay. I should have done one other thing. Now I'm remembering with codominance. Did you do anything in biology with blood type? Okay. Before I go to dihybrid, let me show you a classic blood type situation. Blood type is in here with codominance. Young, you have any questions?
I'm good.
You good? Okay. So, how many blood types do humans have? Well, how many possibilities? Okay, so we're just doing broad types here. So you can have A type, B type, O type. Is there any others? True. Yep. That's getting into the RSH factor, and then AB. Okay. Let's just do the types because if you do the pluses and minuses, which you can, and that's no problem, that's the RH factor. But if you do the pluses and minuses, now that automatically brings it to this level of dihybrid cross. Okay. So this is blood type is an example of codominance. Okay. Now this is the type for phenotype, right? I define phenotype as a physical expression, not in this case. It's not like you can look at somebody's blood and say, oh, that's A-type blood. And then look at somebody else's blood and say, oh, you got O-type blood, I can tell. There's nothing at the physical level that's going to identify this, right? These are all blood proteins. So that has to be tested. But still, we call that phenotype, right? These are the phenotypes, 4 possible phenotypes. Now, what about the genotypes? So this is officially genotypes, right? What are the possibilities? What genotypes could produce A blood? Good. Or this tells you something right away. O must be what? Recessive. Because if you can put that O there and still call this A-type blood, it means O has to be recessive. And indeed it is. Now, but here's an example of codominance with an authentic recessive condition, right? Why is this codominancy?
Because A and B are codominant.
Because A and B are both equally dominant. Excellent. Yeah, there still is a classic recessive, right? Okay. What about B? All right. Or? Yes. O? Yeah, that's the only possibility. AB? Yeah, again, only possibility.
Wait, so why can't A and B, because they're both dominant, so they can't?
Yeah, because you only have two alleles, right? So to have AB blood, you got to have one parent that has that dominant A and one parent that has dominant B, and that's the only possibility.
Yeah.
Same thing here. If you got O-type blood, both parents have given the O allele, the recessive allele. So you know right away, if you've got O-type blood, then your parents, either a parent has O or both parents have the recessive allele.
Yeah.
I have A-type blood. Some of my children have O. I won't get into specifics since you know some of my children, but anyway, not that really matters anyway. But I won't disclose all of these facts for my children. But anyway, some of my children have O. That means automatically my genotype. If I have A-type blood, which automatically my genotype is what? That has to be. Okay. All right, here we go. Let's cross. Let's do the classic example of this. Let's cross a heterozygous A with a heterozygous B and see what happens. You can probably already quickly predict what's going to happen. So this is the situation where one parent is A blood, one parent is B blood, but they could theoretically have 4 children, and each of the 4 children have a different type of blood, theoretically. Right? These are possibilities. Keep that in mind. It's not like, right? It's not this. Oh, our first child was AB. That must mean that our next child could never be AB, right? That's not true. Every child has 1/4 chance of having each of these types of blood. You could end up with a situation of having 4 children, and all 4 children have AB blood. That would be weird, but it could happen. It could happen. I mean, if you're doing that, then you're ending up with the ratio of what? What's the chance that all 4 children might have AB blood? 1/4 times? Yeah. All right, 1/64, right? Is that right? Did I do my math correct? Yeah. Not very likely. Could happen. Okay. We'll get into some of those things later. Okay. Genotype ratio. You got to list each of these. I'm not going to do it just to save time. Genotype, just list each of these individually, right? 1/4. What's the phenotype? 1 AB, 1 B. Oh, maybe I should list these just so you can see the difference of genotype, phenotype. So here's my genotype. That's the genotype. Now watch when I list the phenotype. Tell me the difference. What's the difference? Right. I didn't need to express that recessive O because the type is B, right? The type is B. So when you're listing these for blood type, it looks very similar because between the phenotype and genotype, sometimes you just drop that recessive. That's all you're doing, right? Okay. Any questions there? So how many of you know your blood type? Just curious. Only one person in here knows their blood type? Okay, are you O? Okay. Most, I say most, if you know your blood type, there's a higher percentage that you're O than anything else because O is recessive. This is interesting. And scientists, geneticists have been trying to figure this out for a long time. Do you know what the most prevalent type of blood is in the country and around the world? By a slight margin, but still. Do you know what it is? More people have what? Oh. Wait. More people, there's a higher percentage of people that have O. O is what? That's not supposed to happen. Right? You say the recessive condition is usually eclipsed. It's not as often expressed. But in blood type, it's a little different. For, say, some reason, O is actually the more dominant. Usually, I think in the US, I'd have to look this back up. I think in the US, it's like 51% or something like that. 51% of people are O. Yeah, that is good for O's. Why is that good for O's?
Universal donors.
Right. O's can only donate to O's. And that's why it's good that 50%, right around 50% actually are O, because it's a good sizable percentage, right? Now, did you talk about this in biology, the donation stuff? So O's can only, I'm going to give with the arrow, right? O's can only give to O's. A's can give to, well, I don't need to do that. A's can give to A's or O. B's can give to
B or O.
AB's can give to
Or AB.
AB. Good error. Can AB's give to B or A?
No.
Okay. Cannot. That makes, watch this. Can you give O to an AB though? AB's can only be given to AB's. But could you give an O to an AB?
Yes.
You can.
Why?
Okay. So here's the way it works. This is truly recessive. Now, that means essentially the protein, there's a protein there that is absent. So let's just go through it. This does not have the protein. This has an A protein. This has a B protein. This has an AB protein, both of those, right? So if you give this to this one, there's no protein. Will the immune system in A reject that? No. Will the immune system in B reject that? Will the immune system in AB reject that? No. Okay. So that's why O is the universal donor. You can give it to anybody. Well, then we got to consider the pluses and minuses, the RH factor. But essentially, you can give O to anybody, right? If I give my A to O, what's going to happen? The O person's immune system is going to be like, I do not recognize that A protein. It does not belong to me. And it will fight it, right? It's not mine. That's not my blood protein. If you give B to an O, same thing. If you give A to AB, that's like double proteins, right? O is like, no, I don't recognize A or B. That's why they got to match the blood types when they're doing transfusions. Because if you give somebody a blood protein that doesn't naturally exist in their body, the immune system will fight it for all it's worth, right? That's the job of the immune system. Any questions there? How much time we got? 46. I know this is rapid fire. Sorry, guys. Any questions up to this point? And this is also why I'm recording it, because I have a feeling those who may not be here today, and potentially you guys, again, I'll make this available if you think you need to review back through this, you can. Okay, let's do, so we've done sex linked, we've done that, let's do let's actually, no, let's do dihybrid. Yeah, let's go in order. Let me not break this up too much. Now, here's a dihybrid Punnett square. Just bear with me here for a moment, and then I'll explain what's going on. Notice first, my dihybrid squares, Punnett square is a little bit bigger. I'm going to take that, I'm going to divide it in half vertically, and then each of those two halves in half vertically. Then I'm going to divide it in half horizontally, and do the same, each of those halves in half horizontally. And that means I end up with how many squares? 16. Now, you may not use all of those, and I'll show you why in just a second, but you at least got to start that way. Okay? All right, now, what does it mean to be a dihybrid cross? You are considering two traits at the same time. Let's go back to this. Tall, short. What's another human trait you'd like to deal with here? Freckles. Again, I'm making this very simplistic, right? It's not this easy. Freckles. Is that how you spell freckles? Yeah. Okay. No freckles. So let's cross a tall. Now, notice it's heterozygous, but still tall, right? A tall, freckly person. There's a tall, freckly person. Both of them are heterozygous with, let's just go the opposite extreme, a short, no freckle person. Maybe I should not have chose F because the T's and the F's looks the same. So that might not have been a good letter, but you guys can choose your own letters. I don't care which letters you choose. I chose F because of freckles, but if you want to make that a totally different letter for ease sake, go for it. Here we go. How do we distribute this? This is the biggest difference between monohybrids and dihybrids, is the distribution. Some people call this FOIL or just distributive property. I don't know what you call this. You tell me what you call this in math. I think it's FOIL or distributive property. You take that first allele and you pair it with the first allele of the second trait. You put that here. So capital T, capital F. Then you take the first allele, pair it with the second allele of the second trait. Capital T, lowercase f. And make that lowercase f look really like that. Okay. Then I take the second allele and pair it with the first. And then I take the second with the second. Right, see what I did there? Any questions about what I just did? You're not seeing this, Jan. There you are. No questions? Now, this one is the easy one, right? First with first, but quickly you realize what? What do you realize quick? All the same. Why not save yourself some time, right? In other words, you know what's going to happen here is going to happen what? Here, here, and here, right? The same thing. Now, if you want to do all 16, go for it. No problem. Do it. If you just want to visualize it that way, carry it through. I'm more like, let's cut to the chase and be simple about it. So here's what I'm going to suggest. I'm not doing that. Okay, because I know what's going to happen. So here we go. Let's distribute. Keep the T's together and keep the F's together. Sorry, let me finish this off and then I'll be out of your way. Any questions with that? Now, we still got to do genotype-phenotype ratios. Now, genotype. Any of those other genotypes identical to this one? Okay, so any of them heterozygous and heterozygous? Any identical to this one? Any? Okay, they're all unique, right? In other words, at the genotypic level, they're all unique. So then I just have to list it that way. Now, I'm just going to cut to the chase here. Really, you should be writing the genotypes out. But I'm just going to do that because I'm trying to speed this up a little bit. 1 to 1 to 1 to 1, right? They're all unique. Okay, what about phenotype? Is this one tall or short? Freckly or no freckles? Tall freckle. What about this one? Tall, no freckle.
Short, purple. Nothing for you.
All of these are different too, aren't they? Okay. So here again, I should list this out. I'll list the first one, but I'm not going to list them all. First one is tall, one tall. Now, there's several ways you can do this since it's a dihybrid. This is the way I like to do it. One tall, put a slash between one tall freckles. Right? The second one would be one tall, no freckle. Okay, you get the idea. So 1 to 1 to 1. Again, they're all different. Do you see the difference between the expression of dihybrid and monohybrid? You could do a trihybrid, but then you would have to start with a Punnett square with how many boxes? Yeah, you see why typically we stop with a dihybrid. But you could do a trihybrid cross. You could go on. Distribution gets really crazy. The higher you go, it gets a little crazy. And if you make one mistake somewhere, then it's sometimes hard to figure out where you make a mistake. Okay. So we'll stop with dihybrids. Any question there? All right, the last thing, pedigrees. I'm not sure if you did these I know I did them in mine, but if you didn't have me, did you do the circles and squares and stuff and males and females? Okay. Pedigrees are fun. I like pedigrees. I like all this. It's fun. Anybody still need this? Okay, I'm going to distribute later some worksheets. Again, don't worry, you're going to have plenty of time for these worksheets, but let me take one from the worksheets. That way, I'm giving you something that actually you'll see in just a few moments. Let me do this one. All right, that's a pedigree. Squares are males or females? Which one? Squares are males. Circles are females. First thing to recognize related to pedigrees. Squares are males, circles are females. So in this case, you've got a couple, they have 4 children, 2 sons, 2 daughters, right? What about those that are shaded? Well, those that are shaded, this is the way you define it, they have the trait, they have the condition, or better way to say it, they express it, right? They express the trait, or they express the condition. One job you're going to have is to figure out, is this a dominant or recessive condition? That's one thing. The second is this autosomal or sex-linked? Now, I'll go through those in just a moment, okay? All right, now, first thing to do is just to kind of look at this and assess it. We got 2 children that have the condition. Neither parent expresses it. Do you think that would represent a dominant or a recessive condition? Recessive. The only way neither parent could express it but have 2 children that expresses it is for there to be an allele that is eclipsed. Does that make sense? So if that's true, it doesn't matter what we're dealing with here again, but if that's true, let me just use the T's again. I know automatically that these 2 individuals are homozygous recessive. That also means I know automatically that these 2 parents are heterozygous. Because if they were not heterozygous, there's no possible way they could have 2 children that are homozygous recessive. Now, traditionally, if these two parents were heterozygous, what percent chance would it be for them to have a child that's homozygous recessive? What would that be normally? What percent chance? 25%. We got 50% here. But does that null and void that possibility? Of course not. Because again, you got to remember, that's just 25% chance for every single child, right? Okay. All right. Do we know the genotype of this male here, the son? Now, this is the way you express it if you don't know, right? In other words, he could have got capital T from dad, capital T from mom, could have got capital T from dad and lowercase t from mom, or vice versa. Make sense? We don't know. So you do what you do know, and we do know there's at least one dominant allele. The second allele, we don't know, we just put a dash. We don't know. Okay? Same thing over here. We don't know what this daughter is. Not for certain at least. Okay. Now, by the way, this shows only 2 generations. Some pedigrees, you got 3 or 4 generations to deal with. And then you've got more evidence to look at, and you got more things to consider, right? So this, with just two generations, it's more simplistic, but still. Does that make sense what we're doing here? Is this, okay, here's the next question. We've determined already that this is recessive, right? Not dominant, recessive. You think this is autosomal or sex-linked? Correct, autosomal. Why would this be autosomal and not sex-linked?
Because there's both a male and female that carries recessive traits.
Excellent. Traditionally, right. I mean, there are exceptions to the rule, but those are rare. Traditionally speaking, when you're dealing with a sex-linked condition, you will see males affected. You are not likely to see females affected. So if this was something like hemophilia, we would not expect this, right? This would not be expected. Now, what if this is not the example on your sheet? I'm going to do something for you here so you can see. What if this was the situation? Now, could that be sex-linked? Yes. And if that were sex-linked, this is how we'd have to express it at this point. This is the father. What do we know the mother is? Carrier. So the mother would have to be a carrier. Not seeing that. There you go. What is this son? Yes, he's affected. What is this son? Yeah, normal. What about this daughter? Do we know? Again, so we do what we do know. We do know that. We'll put a dash there. Same thing here. And that's how you complete the pedigree, right? That's how you figure it out. So if we were to classify this, I'm going to say, okay, what's the mode of inheritance? You'll hear that word some. What's the mode of inheritance? Here's the mode of inheritance for this one. Okay, so recessive, right? If, in other words, it takes a recessive allele to have the trait. So you say recessive conditioning, recessive sex-linked. That would be the mode of inheritance, recessive sex-linked. The first one we did would be recessive what? Recessive autosomal. Does that make sense? But this one, as I've changed it, would be recessive sex-linked. Does that make sense? Okay, any questions with anything I've presented? Again, I know it's been rapid fire. And let me pass out worksheets. You're going to have plenty of time. These are not officially the POGIL worksheets. They're different, but I'm going to kind of treat them the same as the POGIL worksheets. That is, you're free to, again, not copy another student, but you're free to work alongside of another student. And please, again, if there's any confusions, let me know. You will, I didn't even set a due date, but something like the end of next week, that'd probably be the due date. So you got some time, but I would just encourage you to start going through it, because when we consider some other things, we kind of add things periodically. It's kind of assuming that as we add things, that you're understanding the concept. There are some on here that say extra credit, so yeah, they are. If you don't want to do them, you don't have to. A little more calculation involved. Did I give you? Okay, oh, you don't want to do it twice? Unless you're gonna, I'll let Joy. I'll let Joy take care of herself for this. Jan, do you have any questions?